Hello. Today we're going to continue our conversation about cellular respiration by talking about step 2, the Krebs cycle, also known as the citric acid cycle. So right before this was glycolysis, which happens in the cytoplasm, and during glycolysis, glucose was split into two 3-carbon molecules called pyruvate. So that's where we're going to pick back up. The pyruvate is going to enter the mitochondria. That's that big orange structure that I've drawn right there. So we'll take a minute and draw that and label it as pyruvate. And then the first thing that's going to happen is that pyruvate actually loses a carbon. So then it becomes a 2-carbon compound called acetic acid. And then let's add in what happened in between there. So it went from having 3 carbons to 2 carbons. Where did that other carbon go? It was actually released into the atmosphere in the form of carbon dioxide. And what was the purpose of that? Just like in glycolysis, anytime we break a bond, we're releasing energy. So there was energy stored right there, and then when we broke that off, we released energy in the form of electrons. This time the electron carrier is called NADH. So we broke off a carbon atom so that we could produce electron carriers. Okay, now let's keep on following this acetic acid through the matrix of the mitochondria. Next, acetic acid is going to combine with a molecule called coenzyme A. I'm not going to draw its structure, I'm just going to draw like a little rectangle. So coenzyme A. And then that gives us a compound called acetyl-CoA. So it has our same 2 carbon atoms bonded to coenzyme A, and it is called acetyl-CoA. No E. Now this acetyl-CoA is getting ready to enter the Krebs cycle. So there is a 4-carbon compound that's already in the Krebs cycle, and that acetyl-CoA is going to combine with the 4-carbon compound, and then we're going to have a total of 6 carbons. So 1, 2, 3, 4, 5, 6. One of them sticking up like that, like it kind of has a funny shape. So acetyl-CoA plus 4 carbons gives us citric acid. Sometimes the Krebs cycle is called the citric acid cycle because the starting compound is citric acid. Then the citric acid is going to go through a series of reactions that removes 2 additional carbon atoms, so it actually returns back to this 4-carbon compound. But all the important stuff happened in the middle of that. So when this is getting broken down, it releases 2 more molecules of carbon dioxide. That's where the carbons went. We exhale it as a waste product. And then what was the purpose of that? Just like earlier, we were breaking a bond to release the energy. So like there's energy stored right there. There's energy stored right there. And when we break the bond, we release energy, this time in the form of ATP and NADH and a new electron carrier called FADH2. So the purpose of the Krebs cycle is to break down pyruvate and be able to produce energy in the form of ATP and electron carriers. And those electron carriers are going to stay in the matrix where they'll be able to power the electron transport chain. And let's label our carbons like we did in photosynthesis. So here is our original 3 carbons of pyruvate. Remember, those came from glucose from photosynthesis. And then our 3 carbons enter the mitochondria, and 1 is broken off and 1 gets exhaled. So 1 carbon was released, making the electron carrier in the process, and then we have 2 carbons still inside the mitochondria. Acetic acid combines with coenzyme A to form acetyl-CoA, so there's our 2 carbons. And then our 2 carbons are going to combine with this 4-carbon molecule to form citric acid. Then the 2 carbons are broken off again and released as CO2 in the atmosphere. So we end up exhaling all of that carbon from photosynthesis, and then guess what happens to it? A plant takes it in and uses it to make more glucose so that we can eat the plant and then break down the glucose to make even more ATP.